Hello to everybody. My name is Diego Luis Medina. I am Associate Professor of Biology at the University of Naples, Federico II in Naples, Italy, but also Associate Investigator and Head of the High Content Screening Facility at the Telethon Institute of Genetics and Medicine at Pozzuoli, Naples. So today I would like to talk about our platform of drug discovery that I entitled Combining High Content Imaging with Repurposing Approaches to Tackle Lysosomal Storage Disease, or LSDs. So basically, the major goal of my laboratory is to study the fundamental biological role of lysosomal function in health and disease conditions, and use this knowledge to identify small molecule correctors of lysosomal storage disorder. In this slide, I try to explain you our modus operandi. So we basically use state-of-the-art cell biology approaches, combining with high content imaging technology in order to investigate different intracellular processes such as lysosomal pathways, autophagic pathways, and the subcellular localization of specific factors that are important for lysosomal biology, and use this as a toolkit of assays that we can use in specific models of lysosomal storage disease in order to phenotype pathogenic pathways in these specific models. Here in the middle panel, you can see several models of lysosomal storage disorders such as Niemann-Pick-C1, Niemann-Pick-C2, CLN3, CLN6, 7, 11, and mucopolysaccharidosis type 3A, that all of them can accumulate specific substrates within the lysosome, such as Philippine 3A that stain cholesterol accumulation in Niemann-Pick-C1 and 2 disease, Shiga toxin reporter that can label specifically globocyte that accumulates in Batten disease, such as CLN3, and the subunit C of mitochondrial ATP synthase, XMAS, that accumulate within the lysosome of various Batten disease models. In mucopolysaccharidosis type 3A, we use an exogenous Prof, the QBSA, that can measure the ability of the lysosome to degrade a specific cargo. In addition to these models, we recently set up in the lab also the Lysotac technology that allows us to immunoprecipitate intact lysosomes from cellular models such as LSD models that I presented before, but also from in vivo using specific tissues such as liver or brain. This technology allows us to isolate the lysosome, and you can see in the immunoblot that we can enrich for lysosomal proteins like LAMP1, LAMP2, and some hydrolases such as cathepsin D in the fraction of the lysosomal immunoprecipitation and compare with the input or the whole tissue. This allows us to identify specific substrates that accumulate in the lysosome of disease model of different lysosomal storage disorder. In addition to that, we also have several mouse models to perform proof of principle in vivo. And in the slide, you can see, for instance, the proximal tubule of kidneys from the mucolipidosis type 4 mouse model. So using these cell biology approaches and specific lysosomal storage disorders models, during these years, we have identified specific pathways in lysosomal storage disorder that can be used or can be targeted in order to identify drugs that can ameliorate the phenotype of this disease. For instance, we identified the pathway of the transcription factor TFEB, which is a transcription factor and a master regulator of lysosomal and autophagic function, and that we demonstrate that its overexpression or activation can promote an amelioration on several of these diseases, such as mucopolysaccharidosis type 3A and Batten disease. So with this approach, we are able not only to get knowledge or generate knowledge from different lysosomal storage disorder models, but also identified pathogenic pathways that can be targeted for future development of small molecule therapies. In addition to that, we combine this platform with small molecule drug screenings, and in particular, we use the repurposing of small molecules that are already approved in the clinics. This allows us to working with what we already know, so with drugs that are already in clinics, and therefore we can move to translation quickly, because we already have a lot of clinical data in humans about metabolism and toxicity of these drugs. So today I would like to put one of these examples as a successful example, in particular for Batten disease. As you know, Batten disease, also known as neuronal ceroid lipofuscinosis, is a group of 13 inherited neurodegenerative disorders that are characterized by the accumulation of lipopigments. Batten disease is the most common of the rare neurodegenerative disorders of children, and is characterized by a rapid and progressive visual loss, cognitive decline, and seizures. Mutation in the CLN3 gene lead to juvenile form of Batten disease. The role of this membrane protein is not entirely clear, but have been involved in autophagy, membrane fusion, vesicular trafficking, arginine transport, and more recently have been shown that it's necessary for exporting lipid metabolism, in particular glycerol phosphodiester or GPDs from the lysosome. Actually, there are no curative treatment that exists for any NCL form, for only one NCL, in particular for the CLN2 disease. There is a FDA EMA approved intracerebroventricular enzyme replace therapy that seems to slow disease progression. So as our modus operandi reveals in the previous slide, we decide to perform a phenotypic screening in CLN3 cells, in particular human retinal pigmented epithelial cells that have been depleted of the CLN3 genes, to reveal a specific lipid accumulation within the lysosome. So we use different reporters of different lipids, for instance, cholera toxin to detect GM1, lipid tox to detect neutral lipids, Shiga toxin to detect globocytes or GB3, and Philippine 3 staining to reveal cholesterol accumulation. We treat human retinal pigmented epithelial cells, wild type, and CLN3 knockouts. As you can see from the different bar plots, CLN3 cells accumulate basically all these lipids, okay? Accumulate them mostly in a spotted localization that suggests a lysosomal localization, but intriguingly, the most significant or the best signal window of such accumulations were related with the accumulation of Shiga toxin and therefore of globocyte, or globotriacyl ceramide, also called GB3. So we decide to study in more detail GB3 accumulation that was not reported before in the scientific literature. And interestingly, we found that GB3 not only accumulating our cellular model of Batten disease, but also in vivo in the brain of CLN3 mouse model. Interestingly, by using colocalization approaches, we found that GB3 accumulates specifically in neurons, but not in astrocytes or microglia. And this is very interesting because it means that this accumulation of GB3 happens specifically in the neuronal compartment of the central nervous system. So after the validation of GB3 accumulation in both cellular and mouse model of Batten disease, we decide to use a cell-based assays to detect GB3 accumulation for a screening of a collection of 1,500 drugs that are clinically approved, are FDA approved, okay? By using this approach and our high-content imaging screening system, we identify around 10 compound hits, and the first two were related with the same category of compounds, tamoxifen and toremifen. This, the fact that we identified two compounds of the same family triggers our curiosity, and we decide to study in more detail the role of these compounds clearing GB3 accumulation in Batten disease models. So first of all, we confirm that tamoxifen reduced GB3 accumulation not only in our genetically engineered cellular line, but also in CLN3 human fibroblasts. As you can see in the slide, fibroblasts from CLN3 patients significantly accumulate GB3 using Shiga toxin reporter in their cells, and tamoxifen significantly reduce the accumulation of this GB3 in these human fibroblasts. As a positive control, we use an inhibitor of the glycosphingolipid synthesis pathway, which is called PDMP. In the blood plot, you can observe the dramatic downregulation of GB3 accumulation in cells treated with tamoxifen. In addition to human fibroblasts affected of CLN3, we also tested neuronal progenitor cells derived from CLN7 patient fibroblasts that were reprogrammed to induce pluripotent stem cells. So you can see here that in these neuronal progenitor cells from a CLN7 neuronal progenitor cell, there is a huge accumulation of GB3 that was dramatically reduced upon tamoxifen treatment. So these results together demonstrate that tamoxifen is able to reduce the pathologic accumulation of globocyte of GB3 in different human models of Batten disease. At this point, we try to delineate the mechanism of action of tamoxifen. Several years ago, I demonstrate that the transcription factor Ibeta, that is a master gene of lysosomal biogenesis and autophagy, is able to induce clearance of pathological accumulation in different models of lysosomal storage disorder by a process that involve lysosomal exocytosis, so the release of the lysosomal content outside the cell. And we published this in Developmental Cell in 2011. So we asked ourselves whether tamoxifen may induce TFEP activation and therefore use this mechanism to clear globocyte accumulation within the lysosome. So in this experiment, we basically use RP19 cells that are knockout for CLN3, and as you can see in control and treated conditions, they accumulate GB3 as expected. The use of tamoxifen in these cells reduce the accumulation of GB3, as we demonstrated previously. But the silencing of TFEB using a small interference RNA depletion restore the accumulation of GB3 even in the presence of tamoxifen, indicating that at least in part, the tamoxifen-mediated clearance of GB3 in CLN3 cells is due to the activation of TFEB. Of course, after these interesting results, mostly in vitro, we decide to generate a proof of principle in vivo by using the mouse model of CLN7 disease. We use this model because better recapitulates in mice the Batten disease neurodegenerative phenotype, and is more and the progression of the disease is more faster than the CLN3 mouse model. So we inject with tamoxifen these mice for 5 months, and then we collected tissues from the central nervous system, so from brain, and we look for different pathogenic markers. And importantly, we found that tamoxifen treatment was able to reduce in the brain the accumulation of GB3 in several regions, such as the cortex, reduce the subunit C of mitochondrial ATP synthase, SMAS, in the cortex and cerebellum, but also downregulate neuroinflammation using the microglia marker EVA1, okay, in the cortex and cerebellum of tamoxifen-treated CLN7 mice. Most importantly, by using the high limb clasping test, we also found that this motor dysfunction that appeared in CLN7 mouse, as you can see here, was rescued by the treatment with tamoxifen. So this data demonstrate that tamoxifen can ameliorate biochemical and behavioral phenotype in vivo in a mouse model of CLN7 disease. So now, as I mentioned before, we demonstrate that tamoxifen induces GB3 clearance in CLN3 and CLN7 disease, most likely or partially through the activation of the transcription factor TFEB, but we know from the literature that tamoxifen is able to target several enzymes and proteins within the cell. So we decide to investigate or identify other mechanisms of action of TFEB that maybe are related with the clearance capacity of tamoxifen in Batten disease. And we do so not only to identify the major pathways that can be activated to ameliorate Batten disease phenotype, but also hopefully identify targets of tamoxifen that are more druggable, and we can perform with them additional drug screenings in order to identify other drugs that can induce the clearance of pathological accumulation within the lysosome in Batten disease. So tamoxifen, we know that activates TFEB because we demonstrate that can translocate TFEB, and without TFEB, the clearance capacity is reduced. We know from literature and many other work and patents that tamoxifen can inhibit PKC and can modulate the estrogen receptor. We know for sure that the estrogen receptors are not involved in the clearance capacity of tamoxifen in Batten disease, because in models of Batten disease where we depleted the estrogen receptor, tamoxifen is able to promote clearance of GB3, but we would like to test other potential targets like the inhibition of glycosphingolipid synthesis pathway by inhibiting glucosylceramide synthase. There are several reports that show that tamoxifen can inhibit this enzyme. And then we would like also to perform a multiomics approach in order to identify potential off targets of tamoxifen that could be beneficial for the effects that we identify in the previous experiment. Regarding lipidomics, we perform a whole lipidomics analysis using tamoxifen in ARP19 CLN3 knockout cells, and we found that compared with the untreated CLN3 cells, tamoxifen can significantly reduce GB3, as you can see in the bar plot, and compare with the CLN3 knockout cells untreated, but also reduce glucosylceramide levels and increase ceramide. This profile of lipids is very typical of an inhibitor of glucosylceramide synthase. So we are now trying to validate this hypothesis in order to delineate whether the tamoxifen-mediated clearance of GB3 is due to the inhibition of glucosylceramide synthase that is upstream the synthesis of globocyte. So it's basically like a substrate reduction therapy. In addition to that, we also are performing lipidomics using our lysosomes technology. So we have a CLN3 mouse model in a background that is express the lysosomes epitope that allow us to take tissues and immunoisolate intact lysosomes. By using this and treating with tamoxifen, we found that, of course, tamoxifen is able to significantly reduce, as you can see in this bar plot, the level of GB3 within the lysosome. This demonstrates formally that tamoxifen reduces pathological accumulation of GB3 in the lysosome. But more interestingly, tamoxifen is also able to reduce the level of GPDs inside the lysosome. And this is very important because recently have been described in a paper that the accumulation of these phospholipids is a hallmark of Batten disease. So we published the first part of this presentation in Embomolecular Medicine, where we define or we identify tamoxifen as a novel small molecule therapy to treat multiple Batten disease, CLN3 and CLN7 phenotype. More recently, we got the approved of the orphan drug designation for tamoxifen to treat multiple Batten disease. And we observed that GB3 storage happens in all Batten disease models that we are screening using a small interference RNA in ARP19 cells. You can see here that all the silencing of all genes related with Batten disease promote the accumulation of globocytes, and that tamoxifen is able to reduce this level in all of these Batten disease models. And we are also working with clinicians in order to determine whether GB3 accumulation is a biomarker of multiple Batten disease. To do so, we are using dried blood spots. That is a very is a non-invasive technique that you use just several drops of blood, so it's really not invasive for the patient. And there we can perform omics, in particular lipidomics or metabolomics, in order to identify the lipids that accumulate in these patients. And we have preliminary data that showed that human patients affected of several Batten disease accumulate GB3 in these blood samples. That is the first proof that GB3 could be used as a biomarker, but also to test the efficacy of novel therapies in vivo in patients with not invasive techniques. We are performing these experiments in collaboration with Angela Schulz and Filippo Santorelli. So the outlook and future perspective of my presentations are summarized in this final slide. So we have demonstrated the validity of phenotypic screening combined with repurposing and high-content imaging technology to identify corrector of Batten disease. The novel pathological accumulation of GB3 have been instrumental to run a small interference RNA screening that identified a set of hits lowering this lipid accumulation, and some top candidates are currently undergoing further validation. So we are using the GB3 assay in order to perform target discovery using small interference RNA screening. The CLN3 knockout lyso-IP technology, both in vitro and in vivo, are now being used by my lab to monitor the effects of specific drugs, such as tamoxifen. And to do so, we use omics analysis in order to identify lipids and metabolites in normal conditions or untreated conditions and after the treatment with a specific compound hits, such as tamoxifen. And now there are several reports that push forward this lyso-IP technology and avoid the use of reporter, in this case TIGEM-192, in order to immunoprecipitate lyso-IP. So there are now ways to isolate the lysosome using specific antibodies against the endogenous TIGEM-192 lysosomal protein. So we are trying that this technology or this approach, because we hope that aligning preclinical and clinical omics-based readouts will facilitate better biomarker-based prediction for clinical outcomes once the treatment reach the patients. And finally, I would like to thank all my previous and current group members that helped me to generate these results and create this high-content imaging screening platform that goes from cell biology approaches to in vivo treatments. I would like also to thank my collaborators, Montera Reimailén, Juan Bolaños, Eduardo Somella, Filippo Santorelli, and Herman Marder-Putten, and all the funding sources. And all of you for your attention. Thank you very much.